Need to worry.
Okay, thank you. Okay, so I should be live. Good morning, United States. Good afternoon, Europe. This time an early afternoon, as the US, like so often, is ahead of us and is already in summertime. We're going to follow the 31st of March. My name is Carsten Schradin, and I'm hosting today's Find. It will be presented by Nigel Bennett. But before we look at today's Find, Lauren Hayes will quickly talk to us about the talk we had last week and the talk we're going to have next week in the Find series.
Hello, everyone. Thanks for that, Karsten. My name is Lauren Hayes. I'm one of the hosts. And first off, we wanted to just acknowledge what happened last week. For the first time, the fine was Zoom bombed. And we just wanted to condemn the content of that without, we just thought it was a horrific thing to happen. And we wanted to condemn that. We did also want to acknowledge the resilience of the speaker. Dominique Defner continued his talk after we had to re-engage, and we really appreciated his willingness to do that. And if you go to the Zoom page, to the YouTube page, I'm sorry, there's actually two links to that. So obviously edited to remove the inappropriate content, but you can go see that talk in two parts. And also just wanted to acknowledge the two people who are leading it. Unfortunately, happened when one of the primary leaders wasn't leading the seminar series. Alba and David were, and just want to let them know we appreciated their resilience to that process. Looking forward, and again, wanted to thank Dominique for that talk. And then looking forward, after Nigel's talk today, we have Kaya Tombak for next week. She's going to be talking about multiple selection forces that shape animal societies. So with that, I'll turn it back over to Karsten.
Okay, thank you, Lauren. So it's a great pleasure today to have as the fine presenter Nigel Bennett. I met Nigel the first time in 2001 when I came down as a young postdoc to South Africa, and he was already then even though he was at that stage still like me, like everyone from us, more than 20 years younger, the silverback of zoology research and behavioral research in South Africa, a very impressive scientist. He is A-rated scientist in South Africa. That means he's rated by the South African National Research Foundation as a world leader, and he clearly is. He's actually British. He did his BSc at the University of Bristol in the UK, but then already came for his PhD to work on mole rats to South Africa, working at the University of Cape Town. And he's for more than 25, I guess, rather nearly 30 years now at the University of Pretoria, a world-leading university in the field of doing behavioral ecology research and behavioral endocrinology in mammals. For example, he's also very good in promoting young scientists. Two young scientists he promoted already presented here at the Find. It was C.P. Machehandle, who gave a fine within the young African researchers. And 2 years ago, Maria Osthuizen was also working together with Nigel, gave another fine talk on the mole rats. So Nigel is an expert on the behavioral endocrinology of mole rats, but he worked also on many other species, including he's been very much involved in the Meerkat project in the Kalahari. He worked with aardvarks, and he worked with so many species, also striped mouse like me. One cannot mention all of them. And he's also, apart from doing basic research interest in conservation, he told me he's very much into helping the conservation projects on the eastern lowland gorillas that are highly endangered and occur in a very difficult war-ridden zone. It's difficult for people to come and help them. Nigel published nearly 500 papers during his career so far. And it's really amazing. He is very productive, and he publishes from specialized journals up to Nature, Science, PNAS, in all the preceding journals. He's a member of the Zoological Society in London, of course, of the Academy of Sciences in South Africa, and many other things, which is way too much to mention. But most important for me is Nigel is a very, very nice person, and he's fascinated by science and by doing research and studying these animals and understanding them. And this is the most positive thing for me from him, because when you hear him talking about his projects, it's really he really takes you with it, and you will realize why what he's doing is so interesting and important. So that's all I can say now, because we don't have enough time, but I'm very much looking forward to your talk, Nigel, and I'm very much looking forward and to hear the news about the mole rats.
Thank you very much, Carsten. Really appreciate it. Just going to share the screen with you a second. There you go. I've got my specialists here helping me. Can everyone see the screen?
Looks good, yes.
Yep. Super. Okay, so today's talk I've entitled Socially Induced Infertility in Highly Social Mole Rats, a tale of two mechanisms of reproductive suppression. What I'd like to do today, before I get on to talk about the mechanisms of social suppression, I'd like to introduce you to the mole rats, the family Physiologically. So African mole rats are basically old world hystricomorphs. Their closest relatives are the hystrix, the porcupines, and the cane rats. And they're also closely related to New World hystricomorphs such as cavies and capybara. They're distributed over a wide range of habitats and soil types. So they're found everywhere in South Africa, from deserts through to the fynbos, but they're characteristically devoid from the Sahel upwards, and you do not find them in the tropical rainforests of Africa, where they are basically dislodged by the genus Tachyrichtes. They have a unique range of social strategies, ranging from extremely solitary dwelling species, in other words, animals which are xenophobic to other individuals, through to highly social or eusocial species, such as the naked mole rat and the Damaraland mole rat. And they show a variable pattern of speciation, chromosomal number, and genetic divergence, which we'll look at as well. So what is fascinating about them? Well, morphologically, they have a very streamlined body. They have reduced ear pinning. They have very small microphthalmic eyes, which they close when they move around in the burrow. They have a low body carriage, and they have those characteristic enlarged extrabutyl incisors, which they use for excavating. Okay. They have a number of physiological adaptations. They have a low resting metabolic rate. They characteristically have low body temperatures, high thermal conductance. The oxygen dissociation curve is shifted to the left, and they have a high red cell pack volume. So they're ideally adapted to living in a burrow system. And the burrow system itself also has unique characteristics. It's slightly hypoxic. It's not extremely hypoxic until you go to the nest. It's hypercapnic. They have high carbon dioxide levels. The humidity in the burrows exceeds 90%, so they have to be very careful when the burrow temperatures go up, because the high humidity and the high temperatures can cause problems of heat dissipation. And they have daily and seasonal temperature profiles which are muted compared to that of the ambient above ground temperatures. Here's our little guys. So there's three species here. These are the three solitary genera. There's Georhicus capensis on the left. Georhicus is a lovely little guy, very colorful. The middle guy is Bathyergus swillus. You can see they've got very well-developed forefeet, which they use for scratch digging. And then you have Heliophobus on the right. Again, a very pretty animal, very furry. That's Georhicus capensis. It's about 300 to 400 grams in body mass. Bathyergus swillus, which can get up to 2 kilograms in body mass. And Heliophobus argenteus, which can get up to about 400, 500 grams. But today I would like to focus my research questions and my research mainly on the cooperatively breeding species. That is the genus Cryptomus on the left, Heterocephalus, which is in the center, and Fucamus on the right. So you all know this cooperative breeding occurs in many species, and a cooperative breeding occurs where more than one pair of individuals exhibit parent-like behavior towards the young of a single nest or of a brood. And likewise, within cooperative breeding, you have helpers, and these are individuals that exhibit parent-like behavior towards the offspring that are not their own or towards the breeder. So they're ideally adapted to looking after a very low number of animals which are responsible for reproduction and procreation. Now, cooperative breeding occurs in a wide range of taxa and also habitats, and there are characteristic features. So the habitat must contain sufficient resources, but often the ecological factors necessitate group living and cooperation. So, for example, in the case of the mole rats, they need to be living in a group where they have to find their food resource. And likewise, many of the other large mammals, such as wild dogs and that, need to have a sufficient number of individuals to bring down prey. Social groups contain dominance hierarchies. The breeders are normally the apex of the dominance hierarchy, and breeding is restricted to the dominant individuals. Non-breeders act as helpers, and groups often contain close relatives, although this is not always true. So here's Cryptus hottentotus. It's a 100 to 150 gram animal on average, and there are 4 subspecies. This is Fucus damarensis, the love of my life. I've worked with them for nearly 4 decades, and I just think they're absolutely beautiful. Again, highly social, we call them eusocial. And then there's a naked mole, which everyone knows about. There's a breeding female on the top. Now, what ecological and phylogenetic factors are involved in the evolution of social behavior in the Bathyergidae? So Ginny Jarvis and I worked for nearly 15 years in a very remote region in Namibia, in central Namibia, in the desert there, called Dordibus. This is a typical characteristic profile of the area for much of the year. As you can see, there's acacia trees. There's very little or any scrub around, just a few bushes. And for most intents and purposes, you can't even see the mounds. They've been eroded by the winds which occur late afternoons. That's the same place after a couple of good rains. As you can see, the mole rats have gone absolutely ballistic. There's huge numbers of mounds which have been pushed up. You can see the leaves of the geophytes. You can see how the mole rats have gone between the geophytes. They're looking for the geophytes blindly, but they actually, once they encounter them, they rework the area. And then you can see to the right, there's a series of mounds that have gone off where they've actually been unproductive in finding mounds, but they've gone off to look for mounds. So the geophytes in these areas are characteristically patchily distributed, and often they're very large. So after many years of research working in the Cape, up in Namibia, and in parts of East Africa, Jenny Jarvis and I and a few other people got together and we put together the aridity food distribution hypothesis. Essentially, this hypothesis posits that solitary species that occur in mesic habitats which can feed on small geophytes, which are abundant and loosely clumped together, do not require you to be social. Also, if you look below it, the rainfall pattern, you'll see for much of the year, the rainfall falls, and it often is above the little dotted line, which is 25 millimeters of rain, which goes down 25 centimeters in the ground. And this actually is enough to promote digging. If you look on the right, you'll see that where the eusocial species occur, or the highly social species occur, there are small geophytes. They tend to be very clumped together. The clumps between these small geophytes are very far apart, and often the geophytes on which the mole rats are feeding upon are larger. So they have to be groups in order to dig these burrows, in order to find this food, when they have the very few times when rainfall events are enough for burrowing to occur. And as you can see on the right side down at the rainfall pattern, there are often very few opportunities when mole rats can dig. And to put you into perspective, a colony of 18 Damaraland mole rats weighing 1.87 kilograms excavated 3 metric tons of soil and excavated a burrow system of a kilometer in just under 2 weeks. So these guys are limited by their digging ability, which usually is aware on the extrabuccal incisors, which they use for excavating. So the hypothesis hinges on 3 factors, and it's being that in the arid regions, geophytes are larger but occur at lower densities. Mole rats forage blindly. There are indications, some people have suggested there are cheromones in the ground, which the mole rats follow, but we haven't actually found any proof of that. So in arid regions, the chance of finding geophytes is increased and the risk of unsuccessful foraging is reduced when you have several individuals searching for a food resource. Now, I think you can appreciate that the energetic cost of burrowing is greatest when soils are dry. That's because the mounds will just cave back into the burrows which they excavate. So mole rats generally tend to limit their digging times to periods when their soil is moistened by the rain, and that restricts the periods during which these animals can dig. Just to show you a molecular phylogeny of the mole rats, and the reason I'm showing you this is to show you a very exciting thing what he found out in 1993. As you can see below, all the solitary species are closely grouped with heterocephalus. The more recent groups are cryptomers. You can see they're the four from Pretoria to Hottentotters. Then the above group are kept as cryptomers because the genus for this group was changed by Koch and Heineck-Bürger in 2006. They actually changed the genus to Fucamus, and with good reason. They don't look the same as the cryptomus species, and they have a variable karyotype. You can see ranging from 40 in the Fucamus machaui up to 74 and 78 in Fucamus damarensis. But the most exciting thing is, as you can see here, eusociality has evolved independently twice within the family, right at the beginning and in the more recent Fucamus damarensis. The molecular clock rates for this were based on fossil-based divergence times for other rodents. There has been more recent phylogenies put together with different aging maps, but for the purpose of this talk, I just wanted to give you an overview. So, heterocephalus is found in Eastern Africa, in Ethiopia, Somalia, Kenya, and Djibouti. Heliophobia is found mainly in the East African regions, Tanzania down into Mozambique, Malawi, and approaching Burundi. Bathyergus is a strictly coastal species that occurs up in tongues into Namibia, but is predominantly found in the coastal coarse sands of South Africa. Then we have Georicus, which occurs sympatrically with Bathyergus and much of its distributional range, but then you have two distributions here, where there are relict populations of Protea on high-level ground. Then you have the genus Cryptomus, which has a ubiquitous distribution in the whole of Republic of South Africa, tongues into Zimbabwe, and potentially could occur in Mozambique, sorry. And then we have Fucamus. The Fucamus has a very wide distribution from the very arid regions of the Kalahari up into the grasslands of the Democratic Republic of Congo. And then we've got these relict populations such as Fucamus zechi up in Ghana area, and Fucamus occasinarius and foxy in the Central African Republic and down into the Sudan. If we look at the evolutionary process, about 40 to 48 million years ago, it's believed that the genus Heterocephalus started to arise. Then speciation took place with Heliophobia arising from Heterocephalus, migrating down south. Then we had rifting events. And then we had, around about 14 to 19 million years ago, we had the progenitors or the early ancestors of Bathyergus and Georicus moving down into the Cape region and their southwestern aridity corridor. About 11 million years ago, we had Cryptomus basically taking over much of South Africa. They occur sympatrically with Bathyergus and Georicus. More rifting took place. Then we have Fucamus migrating up into the African tropical regions, grasslands of the Congo, into the forested regions of the Momiombo woodlands of Malawi, Zimbabwe, and Zambia. And then we had further rifting, cutting off some of the Heliophobius, which had migrated across the African Rift Valley. Now they're stopped, they're prevented from moving back. So the results suggest a complex pattern of adaptive radiation and speciation in the Bathyergidae. We believe that the early divergence of the main clades, that being Heterocephalus and Heliophobia, were probably independent of rifting, but later the other genera, which are Georicus, Bathyergus, and Cryptomus and Fucamus, were probably affected by the cladogenesis from activity from rifting, volcanic activity, vegetation, and climatic changes. We think that possibly what happened with Fucamus is that the river systems may have resulted in a number of speciation events there. So naked mole rats and Damaraland mole rats are divergent, suggesting that eusociality has evolved independently twice in these two species, and these two are the species I'm going to concentrate when we look at the mechanism of socially induced infertility. And comparative analysis by Chris Fawkes in 1997 showed a very strong and significant relationship between the ecological factors, that being food and rainfall, and sociality and group living, supporting the food heredity distribution hypothesis. There are other hypotheses floating around about the evolution of the Bathyergidae, which I won't go into for the sake of time, but I tend to find the hereditary food distribution hypothesis the most plausible. Now, if we look at the relatedness of the individuals in heterocephalous colonies, Fawkes and Berland and O'Brien found that using DNA, multi-locus DNA fingerprinting, that the degree of relatedness amongst individuals in colonies was an estimated 0.81. Now, if you consider that monozygotic twins are related by a factor of 1, first order relatives are 0.5 and second order relatives are 0.25, this suggests that possibly one of the principal causes of the evolution of sociality in heterocephalus could have been the high level of relatedness which occurs in the naked mole rat. But later on, I'm going to show you some more recent work by other scientists, Ingram and Stan Bourdain, and that actually showed that there are normal levels of relatedness in the naked mole rat equivalent to first order relatives. So I think what happened is here that when these first colonies were captured in Tito Ende, they were a population of heterocephalus which were isolated off in a small region and separated from a major population group by a river, and they may be slightly inbred. But this is interesting for later on when we talk about the different mechanisms of social suppression. Then if we look at the colony relatedness of the in Damarensis, because Damarensis, if we look at the mean colony relatedness, we find that the relatedness is a 0.5. Mother offspring pairs are about 0.52. Damaraland females about 0.5. Males 0.46. This makes sense if you've got migrating males into a colony. And breeding pairs are basically unrelated, and then the relatedness amongst colonies is basically 0 as well. But let's get on to the more exciting part of the talk, which I want to talk to you about, and that is the social systems and the mechanisms of social suppression arising in the different colonies and species. So within social cooperatively breeding animals, you have a queen or a breeding female. You can have 1 to 3 breeding males, and then the colonies normally consist of non-reproductive males, non-reproductive females, and juveniles, which act as a workforce. But why do non-breeding male and female mole rats not breed in the confines of the colony? What could this be due to? Well, going back to basics and to your basic knowledge of reproductive physiology, you know that you have the hypothalamus, which has the GnRH neurons. You have the anterior pituitary. Anterior pituitary secretes 2 different types of luteotroph, that's LH and FSH, and these act on the testes and ovaries, bringing about spermatogenesis and ovarian activity. Now, socially induced infertility can arise from 2 different components. You can have behavioural suppression and you can have physiological suppression. Now, in behavioural suppression, mating does not occur, but everything else is operational. So you can see your hypothalamic-pituitary-gonadal axis is functional. So you should expect similar levels of hormones amongst breeders and non-breeders. And the only reason you get a avoidance of behavioral reproductive inhibition is due to incest avoidance and mating interruption. There's no physiological component there. But when you have physiological suppression, you can have coitus taking place. Mating can occur, but if you look at the hypothalamic-pituitary-gonadal axis, you normally find that at the hypothalamic region and at the pituitary region, there is disruption at this particular axis. So how do we know what type of type is operational? And for this, we look at a pituitary sensitivity test. And for this, you take an initial blood sample. You then inject the animal with GnRH. And 20 minutes later, you take a second blood sample and you measure the plasma luteinizing hormone. This gives you an indication of the activation of the pituitary gland from the hypothalamus. By acting, you're giving basically, you're short-circuiting the GnRH system up in the hypothalamic region. And for this, we used a mouse Leydig cell assay, which measures testosterone based on mouse Leydig cells, because it was very, very difficult to find an LH assay that works in mole rats. So we use the bioactive LH. So this LH we're measuring is biologically active. It's not structural LH. And if you look at these two papers, which I wrote back in the 1990s, you can see in the Mashona mole rat, there is purely a behavioral mechanism of suppression. It's purely incest avoidance. How can we tell this? If you look on the left, this is the reproductive female and non-reproductive female given a saline injection. That's the control. It's the vehicle used in which we dissolve the GnRH. There is or should be no or little change in the circulating levels of LH, which is what we get. When we challenge the breeding female, you can see the breeding female's GnRH, after she's been challenged with GnRH, her levels rise. This is reduced from the pituitary lutein. And also, again, in the non-reproductive females, again, we get elevated levels of luteinizing hormone, showing that there's zero inhibition of the pituitary at the level of the hypothalamus. But when we look at the Damaraland mole rat, you can see you've got a completely different picture. If you look at the reproductive females, yes, you get that rise in level after a GnRH challenge on the right there. The saline controls are on the left. But look at the non-reproductives. The response to the GnRH challenge is very low. And this shows that the Damaraland mole rat has a physiological component to reproductive suppression. Yes, they also have a strong incest avoidance component, but there is two components to socially induced infertility in the Damaraland mole rat. And this same picture we see on the right here in the Damaraland mole rat, we also find in the naked mole rat, and we find in the Highveld mole rat. And the picture we see on the left of the Mashona mole rat, we find for the giant mole rats, and we find for the Natal mole rat, where there's purely an incest component to avoidance of reproduction. So let's look at female vital statistics. As I said to you, we're going to concentrate on two specific species of mole rat, the naked mole rat and the Damaraland mole rat. In the naked mole rat, females are physiologically suppressed. If we look at the ovaries, there are little or no follicles being developed. You find the odd follicular development in some of the early, higher, more ranked subordinate females, but most females in the colony are prepubescent, have prepubescent ovaries of little or no follicular development. There are no corpora lutea of ovulation or of pregnancy. The low levels of plasma are much lower than the breeding female. They have low or non-detectable circulating progesterone levels when compared to the breeding female, and they have low or non-detectable circulating levels of estrogen in the breeding female. The Damaraland mole rats also have physiological suppression, but there's a lot more interesting things going on here. So if we look at the ovaries of non-reproductive females, their ovaries show different levels of follicular development. You find primaries, primordial, secondary, even tertiary follicles development, and rarely you find Graafian follicles. But these tertiary follicles and Graafian follicles undergo luteinization. So they form what is called unruptured luteinized follicles. So there are no corpora lutea of ovulation and no corpora lutea of pregnancy, but you get these unruptured luteinized follicles. How do we know that? Well, if you use an immunohistochemical staining, we used an assay to measure 3-beta-hydroxysteroid dehydrogenase in the follicles of these unruptured luteinized follicles, and found that they house this enzyme which is used to convert pregnenolone to progesterone in a two-step isomerase reaction. If you look at the low levels, we also find there's low levels of circulating LH, very similar to the naked mole rat, but we have measurable levels of progesterone, higher than that found in the naked mole rat, and we have some measurable levels of estrogen, much higher than the naked mole rat, but also still lower than the breeding females. And this is because you've got greater follicular development in these animals. If we look at a pattern, this comes from Chris Fawkes's work. If you look at the patterns here, you can see in subordinate non-reproductive females, the progesterone levels are zero. There's nothing going on here. There's no follicular ovarian development taking place. If you look on the right, the queens, you can see there's regular cycles taking place. Again, let's look at non-breeders which have been removed. So non-breeders that are left in the colony, low levels of progesterone, pair them with a male, and they start to ovulate. Lower down, in a colony, low levels of progesterone, housed singly, they start to cycle, and in house of a male, they start to cycle again and eventually become pregnant. So here we know that there's a spontaneous method of ovulation taking place here. Just to give you an idea, that's a typical uterine tract and ovaries of breeding females above the naked mole rats and non-breeders below. You can see that the levels of development in the two groups are significantly different. But what about males? So naked mole rat males, the non-breeders, and this is very unusual, they are the only animals in which non-breeders are physiologically suppressed. They are azoospermic, in other words, they have very few sperm. The sperm are usually non-motile. Very few of them are motile, so non-motile majority. They have low circulating LH plasma levels, much lower than the breeding male, and they have low testosterone concentrations, much lower than the breeding male. If we look at the Damaraland mole rat, though, we have a complete opposite picture here. Non-breeders are not physiologically suppressed. They're oligospermic. They have a number of sperm, but the sperm they produce have increased numbers of abnormalities. And if you look at the circulating basal LH and the response of a GnRH challenge to LH, so GnRH, sorry, you find that the circulating levels of LH are equivalent to breeders. And if you look at circulating testosterone levels, they're normally equivalent to breeders or can sometimes be higher. So again, taking from Chris Falk's work in the past, this was a slide showing non-breeders having majority of low sperm motility, if not any, breeding males, high sperm motility, and then looking at the numbers of sperm, very few sperm, azoospermic conditions in the naked mole rat there. So what factors could be mediating socially induced infertility in the naked mole rat? Could it be a primer pheromone? In other words, could it be a pheromone produced in the nest which blocks blocks reproduction in these subordinate non-breeding males and females? Or could it be a behaviorally induced stress? If we look at naked mole rat colonies, and if you observe them, you'll see that there are a lot more aggressive interactions in naked mole rat colonies than you find in Damaraland mole rat colonies. The alpha female or queen often pulls, tugs, or shoves at the non-reproductives, often passes over them. And you'll even notice that some of the higher ranking females pass over the subordinates much more frequently than the other individuals. So showing on the left here, you'll see that this is a number of shoves per hour. This is actually quite high levels of shoving because we don't see this so much in the mole rats we have. But you can see females are shoved a lot more than males, which are shoved a lot more than neonates and that. So what we have here is that we have a queen that is basically using aggressive or despotic activity to control reproduction in subordinates. Here you see you have a female here that normally succeeds the queen after there's been some type of altercation, but the majority of females have very low levels of testosterone in their dominance rank. So naked mole rats show extreme but reversible inhibition of reproductive physiology in both males and females if removed from the colony. The breeding queen naked mole rat exerts a dominant control method of suppression, but the mechanisms of reproductive suppression in African mole rats are correlated with the constraints on dispersal and also the mating system. So earlier, as I said to you, naked mole rats tend to have, or were found to have, quite high degrees of relatedness. And that may explain why they can have queen succession from within. Whereas in the Damaraland mole rat, you've got a complete opposite situation. If we look at the Damaraland mole rat, microsatellite markers have revealed that the breeding animals in wild colonies are indeed unrelated, a relationship of 0.02. Mean colony relatedness is about 0.46. Mother-offspring relatedness is about that for the first-order relatives, expected for, as I said, for a diploid first-order relative. And this finding demonstrates that normal familial levels of relatedness are also sufficient for the occurrence of eusociated to occur in a mammal. So in other words, that high level of relatedness found in the naked mole rat is not a prerequisite for those animals to become eusocial. It can also occur in animals which have a lower degrees of relatedness. Now, when we started to look at microsatellite markers to investigate parentage in the wild-caught Damaraland mole rats, we were quite puzzled, because not only did we find multiple paternity in colonies, but we found unidentified paternity, suggesting that some males had gone into the colony, mated with the queen, and then exited. So we do find immigrants into colonies of both sexes, but it's predominantly male. So why then would we have this situation? Well, we know that Damaraland mole rats have a high degree of outbreeding. They outbreed. There's a very low level, if any, of inbreeding avoidance alone. The breeding female somehow needs to suppress the non-reproductive females when you get these periods during dispersal when unrelated males can enter the colony. They can come into the colony, breed with an unrelated non-breeding female, and upset the colony cohesion. So multiple queening is prevented by the physiological suppression orchestrated by the queen. So just to show you, we have a very interesting pattern, because if you see here, this is levels of progesterone. Here you'll see in non-reproductive females, progesterone levels are very low. When we removed non-reproductive females and housed them on their own, the reproductive females' progesterone levels rose. And obviously when reproductive females started to go through cycles, their progesterone levels rose even higher. So you can see here, and we did some experimental stuff. Christo Snyman published in 2006 a paper showing that dispersing non-breeding females actually show a spontaneous pattern of ovulation. Now, this is intriguing because much later, Connie Voigt she was trying to unravel the estrogen levels and progesterone levels of queens in intact colonies to try to work out their cycle, the follicular and luteal phase of the cycle. We couldn't get that to work. And the reason we think there is because in the colony situation, the Damaraland mole rat starts to show an induced pattern of ovulation. And it's not unusual for animals to show two different patterns of ovulation within a species. Humans are well known as spontaneous ovulators, but on first dates, some females fall prey to undergoing induced ovulation if they have sexual relations on their first date when they supposedly shouldn't have ovulate. So it can even happen amongst other things. There are a number of lower species as well. A lot of rodents have been shown to show both patterns of ovulation. So it's not set in stone. So what we did is to further investigate what happens when you remove the queen and look at the pituitary sensitivity. We went again to look at we needed to use a method of challenging the pituitary and the hypothalamus to find out what is happening to try and unravel pathways. So as I said to you before, you take a blood sample, you then inject with a GnRH analog, that's GnRH1. You wait 20 minutes to get a maximal response, and then you take a blood sample again, and then you use the bioactive LH bioassay. So we looked at different concentrations of the analog to see what patterns we would get. And as you can see, we actually did, I'm not going to talk about all of this because we haven't got time for it, but as you can see, irrespective of the dosage of GnRH you give, you get the same response in non-reproductive females and reproductive females. So we chose to go with the high dosage. Now, this is a very interesting experiment which we undertook. So we wanted to find out what happens when you remove the reproductive female from a colony of Damaraland mole rats. So here on the left, where we have reproductive female present, there's the pre and post saline challenge showing the vehicle of injection. Saline has no effect. In the middle, we have the pre-GnRH LH level and the post-GnRH level for the non-reproductive females when the queen is present. And then look what happens 2 years later, once the non-reproductive females have been left in the colony situation, but without the queen. You can see they rose, the levels rose there. But because of the strict incest avoidance, they didn't undergo breeding. So you can see that the queen has some effect on the presence of the concentration of LH that's produced. We then went on to try to work out why non-reproductive females and reproductive females have different levels of GnRH. Is it because of the structurally numbers of neurons or pericardia that are present? So we first of all took the whole pituitary of a number of reproductive females and a number of non-reproductive females, and we measured the entire LH concentration in these pituitaries. And as you can see, the breeding females have high levels of pituitary LH, non-reproductive females have lower. Then we looked at the different types of region, the immunoactive GnRH pericardia or neurons in these different regions. And as you can see, in reproductive females, remove non-reproductive females, and non-reproductive females, all of them have much the same in the total brain region, the preoptic area, and the medial basal hypothalamus. And when we look at the mean GnRH soma region, this is the biosynthetic region of the cell, you can see the size of the biosynthetic area is much the same in all three neurons. And if we even look at the different types of GnRH immunoactive neurons in the brains of the three different groups, you can see there's not much difference in the number of non-polar, monopolar, and bipolar neurons. So what we can see here is structurally there's no difference between a breeding female, a non-breeding female, or a removed non-reproductive female. When we went on to look at the GnRH concentrations, we got a complete surprise. I've put the pituitary there again to show you. Look in the reproductive female, the levels of brain GnRH are much lower than the levels of brain GnRH in non-reproductive females. And this at first seems to be an anomaly. Well, actually it's not, because if you've got an animal that's constantly releasing GnRH through the neural pulse generator, you would expect there to be less GnRH present in the brain than in an animal where there is storage of GnRH, but it's not released from the neurons. And this is what Andrew Molteno found in non-reproductive females. He found all these beads of GnRH stored in the GnRH pericardia. You can see them all in the different neurons there. This led us on to try to unravel what is causing suppression at the GnRH level in the neuron of the non-breeders. Now, there are a number of neurosecretory cells that can cause suppression of reproduction. You can have gamma-aminobutyric acid, noradrenaline, endogenous opioid peptides, dopamine, and more recently, you can have RFamide peptide 3. So we decided initially there was quite a lot of evidence that mu-opioid peptides were involved in the suppression of reproduction. It's been found to occur in humans. So what we did, we took a naloxone, which is a opioid antagonist, to try to block the receptors and try to release the GnRH from the neurons in non-breeders. We wanted to see basically if mu-opioid peptides were involved in the suppression of reproduction. So we used different concentrations and we used a different bioassay method. So in the first, we used a single 500 microgram naloxone challenge. So here on the left, you can see there are overectomized. We use overectomized animals because we wanted to avoid the estrogen feedback, which complicates the whole picture of when you start doing work at the level of the pituitary. So we used overectomized non-reproductive females, and here you can see this was for the saline, and overectomized reproductive females with saline here. You can see there's very little change in the difference of the saline injection. But look, when we use naloxone on the right, there was zero change in the non-reproductive females, but there was a slight rise in the reproductive females. We then went on to just concentrate on the non-reproductive females with a long procedure. So what we did, we initially measured the basal LH in the non-reproductive females. Then we did a GnRH challenge, which you can see on the top there. Then we did 8 hourly injections of saline. And then we bled the animal again and measured the LH. As you can see, for the saline control, there's no difference. If you go to the right, we did 8 hourly injections. We had the basal LH. We have the GnRH challenge. Then we have 8 hourly injections of 500 micrograms of naloxone. And if anything, you can see there on the right that the level of LH went down, not up. And a similar pattern occurred if you look at the lower graph. We did hourly injections with a lower dose of naloxone over a 10-hour period, and we got much the same pattern as we had in the above. So from this, we could show quite conclusively that in non-reproductive females, mu-opioid peptides don't appear to be causing any social suppression. Now, as many of you probably know, that the ACTH and mu-opioid peptides are cleaved from a hormone called a melanocorticotrophic hormone. It's a hormone which is cleaved to produce ACTH and also opiates. So we wondered whether cortisol might be involved in suppression of reproduction. Now this, I'm not going to say too much about it because again, the levels were not much higher between reproductive females and non-reproductive females of each sex. And some of you may not agree with me, but I'm actually not convinced that cortisol or corticosteroid is really a good measure of stress. I actually think it's a measure of metabolism. It's a metabolic hormone because it's used to mobilize glucose. And what I've done in the past is Andrew Young and I did some work down in the Kalahari on Damaraland mole rat colonies. We actually measured animals' cortisol during the dry period and during the period of the rains. And we found that during the rains, the cortisol levels in those animals were elevated compared to those in the dry season when they weren't dispersing. Now, what do we know about animals that disperse? They need increased elevated levels of energy for doing the digging. And maybe cortisol actually is a measure of metabolic activity rather than stress. I probably put my neck on the chopping block, but I do wonder sometimes whether glucocorticoids are actually a good measure for lots of things. But then to finally get on to the final component in the Damaraland mole rat, we did some really exciting work. This is the work of Connie Voigt. She'd done this in Germany at Manfred Gar's lab and also in collaboration with Stefan Leitner. And what she did was she looked at the kisspeptin expression, gene expression, RNA gene expression in its in situ hybridization in the brains of breeding non-reproductive breeding Damaraland mole rats and non-breeding Damaraland mole rats. And she further went on to look at other components as well. So if I can just go through this little pattern with you. As you can see on this diagram, there are purple kisspeptin neurons going up to the preoptic area from the arcuate nucleus in the brain. Now, these kisspeptin neurons are controlled further down here by what is called a candy neuron. Now, a candy neuron secretes kisspeptin, neurokinin B, and dynorphin. Now, if you want to release GnRH from the preoptic area into the medial basal hypothalamus, you need to activate that kisspeptin pathway. Now, Connie showed in females that if you look at the anterior ventral periventricular nucleus, in breeders and non-breeders, there's very little change in the expression of kisspeptin in KIS1 RNA activity here. But if you look in the caudal arcuate nucleus, look at the breeders. It lights up like a Christmas tree. Here, there's a huge amount of expression, whereas in the non-breeders, there's very little expression. So we can show quite conclusively that in breeders, there is an activation of the kisspeptin neuron region, and in non-breeders, very little. Again, Connie looked at two other components. She looked at the two gene expressions, P dynorphin and TAC3, that's for neurokinin B, looking at the messenger RNA in the in situ hybridization in the arcuate nucleus. And if you look at the breeders, look at dynorphin, there's hardly any staining in the lower part of the arcuate nucleus. When you look at the non-breeder, the dynorphin stains up very heavily. If you look at neurokinin B, we get the quite opposite effect. In the arc, you've got very high staining in the arcuate nucleus, but in the non-breeder, very little. Now, these candy neurons are suggested to be the region of the neural pulse generator. So when you have neurokinin B being expressed and dynorphin is down, as what you find in breeders, kisspeptin is released, and that activates the GnRH neuronal system. In non-breeders, we find kisspeptin is low, neurokinin B is low, and dynorphin levels are high. So this suggests that in the non-reproductive females, the kisspeptin pathway is switched off in non-breeders. There is also another component which we need to look at, and that is another molecule, another RFamide peptide called RFRP-amide. And this is important in inhibition of reproduction. Now, in breeders, if you look at the PVH, the dorsal medial hypothalamus and the third ventricular dorsal medial hypothalamus, if you look in breeders, you can see that the staining up in these three regions is not so great. But if you look lower down in the non-breeders, in the last two panels, there's a lot more lighting up in the dorsal medial hypothalamus and the third ventricle of the dorsal medial hypothalamus for this RFamide-free peptide. So in non-reproductive female Damaraland mole rats, GnRH secretion appears to be due to an inhibition to an inadequate secretion of the peptide kisspeptin. And we believe that this is exhibited by a reduction or a lack of staining of kisspeptin in the AVPV and more so in the ARC areas of non-reproductive females. The results revealed very little evidence to suggest that beta endorphins play a role in the suppression of GnRH secretion in non-reproductive females. So in essence, kisspeptin erectus staining occurs more prominently in the breeding females of the queens, and particularly in the arcuate nucleus, but seems to be much lower in the socially suppressed non-reproductive female subordinates. In males, we had a very interesting thing. Connie went on to look at the kiss in males, looking at the caudal arcuate nucleus. And as you can see, if you look at breeding males, there's just slightly more staining in the breeders for KISS1 than there is in non-breeders, but not greatly so. But look at the RFRP MRA component lower down. There's much more staining in the dorsal medial hypothalamus there than there is in the breeder. So again, these two hormones are incredibly important in being involved in the suppression of reproduction in mole rats. Now, this is the last part I want to just concentrate on. That is the role of prolactin. Now, Jenny Jarvis and I, we often used to laugh because when we looked at naked mole rat colonies, I once said to her, Jenny, why does the non-reproductive mouse have such prominent teats? And she said, I've got no idea, Nigel. And I said, Jenny, do you not think they've got prolactin? And we had several people try to measure prolactin. We sent samples to Alan McNeely's lab up in Edinburgh, and they couldn't measure prolactin levels in the mole rats. It was only with Andre Hunsvint's lab, the endocrine research laboratory at UP, that we decided to import some kits from China, the guinea pig prolactin kits, and we validated these kits for mole rat samples. And as you can see, very, very interestingly, if you look at Fucumus damarensis, the levels of prolactin there in the circulating plasma are incredibly low, if not non-detectable. And as you'd expect in a queen, which periodically shows cycling, you have sometimes elevated levels of prolactin. That was what you'd expect in a normal female that undergoes reproduction. But look in the naked mole rat. In the queen, the non-breeding female, the non-breeding male, and even the breeding male, the levels of prolactin are highly elevated. In fact, you have hyperprolactemia occurring in many of these animals. Now, as we know, prolactin is a very important hormone in cooperative breeding in birds. It's been found to be very important in birds, and more recently, it's been found to be very important in marmoset monkeys. The work of Ziegler and Abbott and that have shown prolactin to be very important there. But also prolactin, high levels of prolactin inhibit FSH and LH. So we have here a very interesting situation. We have here animals which have high levels of prolactin, which switches off reproduction, but we have high levels of cooperative care in these animals as well. So I'm going to end off with this because the naked mole rat, to me, is a fascinating model to further investigate this. I don't want to talk more about this because we've got some very exciting research that we've been working on now, which some of it is about to be published and others is still in the later stages of being undertaken. But to us, it seems very interesting that we have this taking place. And in fact, we have one genus of mole rats, which we have the whole spectrum from high levels of prolactin in one subspecies to zero non-existent levels in a species which shows pure incest avoidance, to some where there is an intermediate level of incest avoidance and social suppression. So watch this space for the future. I've only got 2.5 more years left in my research field, but I'm sure that the people that come after me are going to take this to the new level. Unfortunately, we can't go further than this at present, but I think I was talking to Facundo today and I was explaining to him how to try to do this type of work in an exotic model on your own is incredibly difficult. I mean, if you think about people that have been looking at rats and mice, there are massive research groups going into this reproductive biology and looking at suppression of reproduction and creating mutant strains of mice, which they can try to unravel components of reproductive inhibition in the mice. When you work on a species which is social, and you work on a species which is very difficult to catch and also very complicated to keep in the laboratory, I think we've unraveled quite a lot of stuff here. And it still needs work. The naked mole rat and the Damaraland mole rat both do not show any obvious response to opioid peptide suppression, but there is this dichotomy with the prolactin levels here in the two species. So something must be happening with the prolactin in the naked mole rat, which is not happening in the Damaraland mole rat. So I leave that to you, and I just would like to thank you. I'm sure I'm going to get some people say to me, well, I don't think your mole rats are actually eusocial. I've argued this for a very long time. I go by the definition of Michener, who says that there are three components to the definition, which the naked mole rat and the Damaraland mole rat confer to, and also of Paul Sherman's spectrum of continuum of eusociality, where both the Damaraland mole rat and the naked mole rat hit the extreme end of the eusociality spectrum. And as we've noticed in more recent years, there have been more and more invertebrates have joined the eusocial club: shrimps, beetles, aphids that have joined the Hymenoptera and the Isoptera. And I don't think there's any reason why we should exclude some vertebrates from not being placed into that position. Otherwise, we've got to completely define a new definition of these highly social systems which occur in mammals and birds, because I think there really is some fascinating stuff here. But thank you all very much for listening to the talk. I'm very grateful for you to spend your time with me and listen to some of the work I've done. I had to try and steer away from sociality because, as and excuse the pun, you had Marcus Zottel and Marika Usays and give you two fine talks at Fine. And I had to look at something else which wasn't a duplication of the stuff which we've done already. So thank you very much. And thank you, Carsten.
Yes, thank you very much, Nigel. Very interesting talk. Lots of hormones. That reminds me of my PhD and what I did afterwards, also with the striped mice. Now we come to the question and answers. To remind everyone, if you have a question, then please type a question mark into the chat. I will then call upon you one person after the other. When you're a student, you can put an S for student in brackets behind your name. We will preferably first give the students the opportunity to ask questions. When I call you up, please first introduce yourself quickly, say what's your name, where you're working and what on, and then ask your question. Yeah. And they'll be starting with a question by Theo Baker.
Yeah, hello, I'm Theo Baker from Bakker. We call it from the University of Bonn. I'm retired professor in evolutionary ecology. I had a question on the speciation of the naked mole rats. Starting with the naked mole rat, the speciation is southwards and not westwards. What's the reason for that?
We're not really sure. It could be the aridity of the environment. I mean, you know, we don't know what in the past as to how. I mean, it's supposed to have been very, very lush. All I know that there was, when I've looked back in the over geological time, there was this southwest aridity corridor, but it also opened up grasslands in that area, and it could be that they would attracted to the grassland area and the geophytes which they feed upon. You see, it's quite possible the geophytes, there hasn't been any recording of geophytes west of the region of Kenya and Tanzania now. And we don't know in the past whether it's just because there's been climatic change over time, but geophytes are found very ubiquitously in the southern part of Africa, in the eastern part of Africa and Central Africa, but they disappear as you get into the tropical forests of Central Africa. And we don't know in the past because, you know, I've read that 8,000 years ago, it's believed that the Sahel was actually quite tropical. There are still crocodiles in that region, but there's no fossil evidence of geophytes there. So I think that's what's happened. And also looking at recently, one of my colleagues, Herman Montoya, did a study. He identified something very interesting. He pointed to a reference to me where someone had caught a Bathyergus in Namibia, or found a Bathyergus fossil in Namibia, which had a long tail similar to a naked mole rat. Because we could never work out why naked mole rats have a long tail and all the other Bathyergids have a short, small tail. So it's possible that they migrated down and then they lost, over time the tail has been reduced because of the digging activity, and the naked mole rat has maintained that tail because of the, why it's lost its pelage. So I can't really thoroughly explain why they went south and not west, but I think it might be due to the food resource.
Thank you. Thank you for a very nice
Pleasure.
talk. Yeah.
Good, then we jump forward to the first student question by Laura Miller.
Hello, really nice talk.
Thank you very much.
My main question is, is there ever any variation in ovary development in the non-reproductives seen in other species? And also, I know you mentioned there's a queen, like a single queen, I'm assuming, but
Yeah,
can
there is.
anyone replace the queen? Or...
Yeah, well, in the naked mole rat, you tend to get, they're very unusual because most of the other species of social mole rat have to outbreed. There's a strong incest avoidance component. So what happens is if you look within a colony in the naked mole rat, you find that the queen shows full development of follicular development. They have going from primordial right through to the corpus luteal production. But some females, which are high up in the dominance hierarchy, some of the sisters of the breeding female may show follicular development up to the Graafian follicle stage. And then for some reason, it doesn't go beyond that because they don't get a mating opportunity with the breeding male. But most of the other females, for all intents and purposes, they look like they're prepubescent. They just have primordial follicles, maybe some primary follicles and nothing else. And it's quite striking. So you do get variation amongst that in the naked mole rat. In the Damaraland mole rat and some of the Highveld mole rats, so that there is follicular development and to different degrees, depending on where they are in the dominance hierarchy. But they do show follicular development. I mean, it would be hard to say, I wouldn't like to say to you that lower ranked animals have less follicular development. It's just that they don't show any, very few show a development of a Graafian follicle. It's normally the very big animals, which are probably the ones that are going to disperse.
Thank you.
Pleasure.
Come to a question by Raghavendra.
I'm Raghavendra Gadakar from the Indian Institute of Science. Hello, Nigel. That was absolutely fascinating.
Oh, thank you.
Kept up with your work a little bit, but this was really nice, very, very nice update. I have no difficulty in classifying Damaraland modelets as eusocial, absolutely. I have several questions, but I'll ask one and then come back if there is time later.
Yep, fine.
The question that still remains in my mind is, in all these physiological pathways that you outlined, where does the presence or absence of the queen intervene in the pathway? And especially, where does the absence of mating intervene in this pathway to bring about this differential development or the lack of development? Exactly where does it happen?
You've hit it's a cherry on the top. Chris Fawkes and I, for years, I mean, we've tried to unravel this. And, Chris did a lot of headspace in the naked mole rats stuff. In fact, he got very annoyed because the BBC came to do a film once and Chris said to them, we've been looking at headspace. He said, but we don't find any change in pheromonal composition. And he said, how then does the queen prevent herself from switching herself off? So the BBC took this up and said, oh, there's pheromonal control in the naked mole rat, and that's why they have this skin and no fur, because there's pheromonal spreading. So Chris spent several years doing nest transfer experiments, and he couldn't find anything there. Then he suggested, we chatted, and he said, maybe it's the bullying which occurs, and maybe the stress hormones that are higher. But we've looked at plasma cortisol, we've looked at urinary cortisol, and we've looked at fecal cortisol levels. And there's no difference between queens and subordinates. So that's why I don't really have this great trust in glucocorticoids. And I was hoping now that we might find, with the prolactin, why is it that, how does the queen activate that high level of prolactin in those non-breeders to switch them off? But at the same time, how does the breeding male But if you look, the breeding males do have slightly lower prolactin levels than the non-breeding males. How does he not be switched off? And the queen has also got high levels, but remember, she's cycling all the time. So she may be, at the late stage when she's pregnant, we can't always find out when they're early pregnant or when they've just, we know when they have babies. We sometimes have to take those levels, and those prolactin levels are high. But how does the queen activate the prolactin in them? And that is a difficult question. So you hit it on the nose there. And in the Damaraland mole rats, I've often had people challenge me. They say, Nigel, you're talking nonsense. Those animals are not physiologically suppressed. They simply don't have incest avoidance. But as I showed you, I tried to show you quite meticulously, there is two components to socially induced infertility there. And I spoke to Dave Abbott several years ago, and he said he gets the same thing in his marmoset monkeys. He said there's avoidance of incest. He said, but Nigel, there's a physiological suppression. And he said it's a definite physiological suppression. Now, what is interesting is you might have this pushing by the queen, which would supposedly stress out the other ones, but it doesn't, because cortisol levels are not changing. But for me, having that high prolactin would be a good answer. I mean, it was, why do they have these teats? Males don't feed the babies, but they have these teats. And that's a part of the problem with hyperprolactemia in humans, is that males develop extended nipples and females, their breasts develop. So I think there is something in that. In the Damaraland mole rat, what puzzles me and has puzzled me is that in general, the queen doesn't bully the colony. There's very little dominance in the colony, and the breeding female isn't the dominant animal in the colony, it's the breeding male. So I think the outbreeding is an important component there. But as I said to you, during the period when there's migration into the colony, somehow there has to be some physiological suppression, otherwise you get a disturbance to the colony. And we've shown that there is that component. I mean, it is difficult, but yeah, I'm still trying to unravel that thing. That's the cherry on the top. What actually is it that the queen does, which doesn't affect her, to bring about this social suppression? A lot of people, they try and sort it out quite easily. They say, oh, Damaraland mole rats, they're self-restraint. The non-breeding females simply don't breed because they don't want to. But that goes against all of Darwin's theories about natural selection and sexual selection. Why would you inhibit yourself from breeding? I mean, there must be something operational there. And as I say, I think there's two components, and I think that's what makes it more difficult in the Damaraland mole rat to unravel it. In the naked mole rat, we're still doing some other work on that, and hopefully that will come out soon. But at the end of the day, I think these animals are always going to mystify us. I don't think we're going to find the answer. I mean, look at the bees. Everyone keeps on about the queen pheromone, and they talk about the glands producing these things. But at the end of the day, there's still another component that takes place. The royal jelly is important, and that. So it could be, I mean, Hazel Nichols has recently done some work looking at semiochemistry in the Damaraland mole rat, and has shown that some of the animals have different profiles. It could be there is a pheromone, but I don't believe it is a pheromone. And I think they have an odor, group odor, but I don't think it's a pheromone or a chemical that's volatile that's causing suppression of reproduction. Otherwise, how would the queen prevent herself from doing that? So I think in the case of the naked mole rat, aggression is important, but why she is aggressive, I don't know. Why is it not being manifested with the glucocorticoid concentrations? And how does that prolactin get produced? And in the Damaraland mole rat, how does the queen maintain her dominance in the colony where she's not aggressive, and yet she suppresses all other individuals? I mean, obviously the strong incest avoidance is there, but she has to have some control when these animals migrate into the colony. So yeah, I don't have the answer. I mean, I should have said to you, I don't know, but I'm just explaining to you, it's not a simple question.
Fascinating. Thank you.
Yeah, pleasure.
Yes, I myself, I have a lot of questions, and the most complex I have adds to Raghavendra's question, and I think Nigel, you partly gave the answer already, but I will still phrase it again. You gave this nice overview that this social infertility can come into action by two mechanisms. The first one is simply inbreeding avoidance. And of course, this is a seminar on evolution. And from an evolutionary point of view, one can easily explain an individual has costs when it shows inbreeding, so it doesn't breed to have reserves for later breeding. But then for the physiological suppression, people always tell the story like it's against their will, but I mean, also these mechanisms must have somehow evolved. And in theory, if it would be better for them not to be suppressed, they should evolve resilience. So to some extent, there must also be a benefit for these individuals to be willing for this physiological suppression. And I guess then there must be a conflict at which level they are still suppressed and not between the queen and the other females. Is there anything known or done about this when this trade-off or this conflict is resolved, of course, in the one or the other direction?
I think Tim Clutton-Brook and I think Laurent Keller wrote a couple of papers a few years ago in Tree, basically trying to unwind this theoretical question about what are the benefits and what are the costs in that. And I think the benefits, Carsten, of having group living is, in the case of the mole rats that occur in these arid regions, if they don't live together, they're not going to survive. So the benefit of being in a colonial situation is that they survive because they work as a unit. But the costs to the non-breeders is that they're working to look after the litter of a single female, which again is their relatives, but there must be some internal struggle at some stage for them to want to go off and establish their own colonies. And I think it's at that stage, you know, it's got to do with energy balance and you know, thermoregulatory stuff as well. I mean, if you're an individual that goes off on your own, you've got to have enough energy and you've got to have a mechanism to be able to live on your own and conduct that escape, if you want, from the colony to live that time on your own until you find a partner. Now, you get dispersal morphs. In the naked mole rat, you get a morph which bulks up. It produces more fat and it has higher amounts of brown fat. The same with the Damaraland mole rat. Mike Scantlebury showed that, you know, a paper he did with Nature, that the dispersers bulk up, they get very big, and they build up brown fat in order to be able to disperse. But there's a limitation to this as well, because you've got to have the correct food resources and you've got to have time in which you can do that. Now, in the Damaraland mole rat, where they occur in the central Kalahari, where the rainfall, I've got data for 100 years from Daughtibus, where the average rainfall is 120 millimeters. There, you don't have the opportunity very often to disperse. And when you do have the opportunity to disperse, you've got to make the most of it. And what we found is, Jenny and I were quite surprised, just prior to the rains, I don't know how they do it, the mole rats bulk up. They bulk up and they wait for the rains. Somehow they have an inbuilt mechanism that they're almost anticipating that rains are going to occur. In the case of the naked mole rat, we don't know. Stan Bordes mentioned he's found animals on the surface running around quite often, and I think also Paul Sherman mentioned it. But you know, they have to obviously migrate off, and they have to meet up with another component. And these animals are normally much more friendly than the other individuals in colonies. So it could possibly be that they've got this reduced xenophobic behavior in order to join colonies or to join another animal in order to become a disperser. Now, recently, Marcus Sottle and Jack Forley and Tim and that have commented that in the extreme south of the Kalahari, they found large numbers of mole rats that occur on their own. In fact, large numbers. But now, I'm not sure, but the rainfall there in the last few years has increased dramatically, and the food resource there, which are cucurbits and eland beans, they're large enough to support an animal for a period of time if they're found in a clump. So it could be that under more extreme conditions, favorable conditions, a Damaraland mole rat or a naked mole rat could live on its own for a longer period of time if the food resource is there and it's got the energy balance for it. But in there, as I say, in the more arid distribution of the Damaraland mole rat, we rarely, if ever, find animals on their own. And if they are, they're big animals. They bulked up. They've got a lot of fat reserves, and as I said, there's very few opportunities for them to disperse. So I think what is going to happen, what worries me in the case of the Damaraland mole rat, is in the future, there are big changes taking place in their distributional range. There's increased rainfall in some areas. There's decreased rainfall in the central Kalahari. So it could be that we will get a westward movement of some of the species into areas which are getting higher rainfall regions and regions which get less rainfall, you may get local extinctions because the geophytes go extinct. And to put you in the picture, that has happened in the Northern Cape. There has been, I spoke to a chap up there. He said there's been 32 species of geophyte which have gone extinct because of the low levels of rainfall and because of the mismanagement of land. In such situation, there was the species of solitary mole rat, you probably know about it, the Namakwa mole rat. It's almost gone extinct now in the Northern Cape because of this change, climatic change. And they're moving into areas, they're moving down to River and that type of area where they've got these coastal fogs where the moisture can sustain them. So yeah, it's a difficult, it's an awkward thing to say. Why would they stay together? Why wouldn't they disperse and set up their own colonies? And why would they forego reproduction? And I think what they do is I think they actually realize, I don't know, we've got to be careful we don't become anthropomorphic, but in a mole rat term, I think they realize if they don't help and look after the colony during those dry periods, they're not going to survive. They're going to disappear. So yeah, I think over evolutionary time, this whole concentration of development of sociality has occurred. And as they've moved down into the more mesic regions, it's dissolved. I mean, we see that in the Cape, Georychus has become solitary. It's originally from potentially a social mammal. Bathyergus is solitary. Many of the Heliophobus populations are now solitary. They've become solitary because they didn't need to be social anymore. It's because they migrated into an area where the food resource was enough for them not to require the assistance of other individuals to look after them. But at the same time, they've taken on new costs because now they have to find a mate at a certain time of the year, and they have to develop different acoustic or seismic communication, in other words, to get together to mate again. So yeah, it's difficult. I mean, I'm afraid I'm not into this thing about, there's someone said to me once, mice choose each other based on MHC genes. I don't think they do. I think an animal just grabs another animal, mates with it. I think it's like humans. Often most humans, they don't look at, aspects. They go for it. They look for the person which they like. I mean, I think, I don't know if I've explained everything well there, but I think that the costs of living in a group are much less than the benefits accrued. But when the rains occur, I think there's a swap. I think the costs involved in being in a group probably increase because of antagonism, because there's opportunities to disperse. The benefits become lower because the soil is moist, they can disperse, they can find resources more naturally, and that's probably what triggers them. But I don't know, Carsten, I'm not very theoretical. I think that possibly is the only mechanism I can think of.
Okay, thank you. I mean, from what you said, I think as long as they live in groups, the cause of being physiologically suppressed, this might actually be a benefit because they save energy, which they can then use for dispersal. And when this rare event occurs, use the energy to disperse, settle yourself, and then you can invest into ovulation. And if you manage this, then you're well off.
It's like I mentioned to you, the patterns of ovulation. Like, when you disperse, you are spontaneously ovulating because you want to be able to mate. You want an opportunity to mate whenever you can. Once you're in a colony, why do you need to be spontaneously ovulating? You need more to be induced ovulator because you have your mate there. So you just mate when the male mates with you. You don't need to spontaneously ovulate. So I think that's why there's two patterns. But again, I don't think mole rats are that simple. I mean, when you start taking out the queen to try and collect samples from her, or you isolate a queen off to try and get the urine and that, you're going to interfere with the pattern of what's happening. Even if you keep them in the colony, you've got to pick up, you can only basically sample with urine then or feces, and you're going to get a rough idea of what's happening. I mean, it's very complicated.
Yeah, for future generations of scientists, maybe to mention also the nice thing you mentioned, the Damaraland mole rat has a broad distribution. It's called Damaraland because it occurs there in very dry mesic areas in the Kalahari, then less mesic. I mean, the Kalahari Meerkat Center, they have more than 200 millimeters of rain. They occur at Mayfield Site 2 that has winter rain. So there's a lot of variation in rainfall, in food, in soils. And we can expect it will also be represented in variation in the physiological mechanisms and suppression.
Yeah. I mean, I'm sure, with time, you may find that over many, many centuries that if the rainfall increases in a particular area where there was physiological suppression, it may fall away. There may be no need for it anymore because the animals disperse so regularly. Why do you need physiological suppression? I mean, we may be looking at, the end product in some of the species, may be looking at the end product of something that was there and is declining. We don't know. There's many, there's lots of more research that needs to be done.
Okay, now we move on to a question by Maren Hook.
Hello, yeah, my name is Maren Hook. I'm at the University of Derby, and a long time ago I did my PhD on mustache tamarins. And so I don't really have a question, more of a vague, muddled thought, because you mentioned callitrichots a couple of times.
Yeah.
And I know that callitrichots are often classed as a textbook example of female suppression. And the thing is that all those people working on callitrichots who work in the field are somewhat skeptical of that, because those data that were collected from the field, which were, I think, mostly from different species though, so rather from tamarins than from marmosets, but I think there's also a study on marmosets, and they did not find the suppression of the subordinate females. So in the data are obviously very messy from the field and usually extremely low sample sizes. But the females in mustache tamarins and the females in some other species that were tested in the field were essentially the same as the female. The same for the males. So hormonally, there was no difference whatsoever. And we were always vaguely thinking whether there is an effect of the living conditions in the field versus in the lab. And I don't want to call it an artifact. I think it's just different living conditions. So there might be better possibilities of exchanging hormones that from the breeding female, or less possibilities to avoid if there is aggression going on, which in marmosets and tamarins is on the average day extremely subtle. So as a human, you normally don't notice what's going on, at least not in the field. And so whether there is a difference also between the Damaraland mole rats and the naked mole rats in terms of how, I don't know, how pheromones could be distributed or anything. It is fascinating how these differences occur and that seem to be so flexible.
Yeah. And one thing is, when we did the work on the Damaraland mole rats, we did work in the field initially. I mean, you won't believe this. Jenny Jarvis and I had to put the animals a little bit in the sun because we took samples from the feet, from the vein in the foot. So we had to heat the animals up a little bit when we did our challenges. So we were working in the field, and we'd done all that work in the field. And then we actually repeated it in the laboratory afterwards just to double check. And the same with Andrew Young. When we did the work in the field, we had to bleed and then release the animals. When we caught the whole colony, we had to release them back into the tunnels the next day, because their tunnels often can be taken over within a couple of days. So, it's very, very, you've got to be very, very careful with the Damaraland mole rats. I actually had a situation once. I was in the field and we caught out a complete colony which we'd marked. And as I said to you, in the old days, we didn't have these wonderful pit tags and that. We had to do toe clipping. So we marked them. We had number 1,700s and we had all sorts. So we caught this whole colony. And for a day, there was no trap. No one come to the burrow. The next, and these are actually very interesting, because up where I was trapping in Dordibus, you get black variants and you get fawn variants. So you can get a colony which is entirely black pelt, a colony which is entirely fawn, and you can get a mixed colony. So we had this colony which we knew, and then the next day we started getting fawn animals come through, which were marked the 700s group. So within a day, those animals had taken over the burrow of the other group. So you have to be incredibly careful. And in mole rats, you have, mole rats have a dominance neural phenotype. And it's possible, I suppose, that in your marmosets, you don't have that type of thing. could be that that's one of the explanations. But I did find it strange that Dave Abbott was, when I talked to him, he just said to me, yeah, we've got two components of suppression in the marmoset monkeys. And I found that quite strange because he's worked on marmosets for quite a long time. I think he was actually at Wisconsin, the primate center there, wasn't he? That's fascinating. But no, you're right. I think one has to be very careful how you interpret things, and you have to marry what you get in the field with what you have in the lab, because you can get lab artifacts as well, which can be quite misleading. But yeah, what causes the difference between the naked mole rat and the Damaraland mole rat? I mean, I think it's going to be a few more years before it's found out. I'd like to be still alive to see what happens when that comes, but I don't think I'll be involved in that. I think it's a bit too much down the line.
Okay, then we continue with another question by Raghavendra.
Yes, I would like to ask another question, if I may. It's not every
Yep.
day I get a chance to talk to Nigel. In the beginning of your talk, you showed us some data on the relatedness in the naked mole rats.
Yes.
And then you hinted that this may have been because the population was isolated. And you said later on people have found something else. I didn't quite get that. So
Yes.
do we now have information that the relatedness is not so high?
Yeah, so what happened, the naked mole rats, the initial work which was done on the animals from Tito and Day, Jenny said to me that when they caught the animals there were just 3 or 4 colonies. They were actually on a sandbank, and they caught the colonies out and they transported them back to South Africa. And then obviously the genetics was done in London by Chris, and they had these high levels of relatedness, 0.81, and everyone drooled on this and said, eusociality in the naked mole rat because of a high relatedness, just like with the bees and the wasps. Then I think it was maybe Colleen Ingram and maybe Stan Broaday and some other people. I can't remember the names of them, but they did some later work on some colonies captured down in Savo, and they found that actually there's normal familial levels of relatedness there. So in other words, you don't have this high relatedness that's found in the naked mole rat. But this could explain that this inbreeding, which takes place in the naked mole rat, could be explained why you get this, because they are able to inbreed it at high levels of genetic relatedness, whereas the Damaralands don't. Something that's very interesting now, there was a paper by, I think it was a Mark Freeman from Florida, and this really fascinated me. He took Damaraland mole rats out of the colony and kept them isolated for a couple of months, and he was able to get those animals to inbreed, which I found incredibly fascinating. So, there's something really, really interesting. Why did that incest avoidance break down? And I was absolutely stunned by this, because when we did all our genetic relatedness of our colonies, we always got this 0 degree of relatedness between individuals in the field. We never had that. And then in the lab, we've never had, until very recently, we only ever had 1 queen in a colony. And then a while ago, one of our queens died, of a very old queen. And we had a succession of killings and queens coming up through. And recently, my postdoc, Daniel, was looking in the group and he said, Nigel, he said, I think there's 2 queens in this colony. But this is because they've been maintained in captivity and because there's been such a lot of disturbance. But the one queen has become a queen. She's breeding in that, but the other queen has extended teats and she's perforate, but she hasn't given birth. So we don't know whether she's just a rival queen that's in the waiting, or if it's like some artifact from captivity. But of all the times I've trapped in the field, I've never caught plural queens. And in the laboratory, I've never had more than 1 queen. So this is quite unusual. It's very, very unusual. So again, yeah, I mean, so this relatedness in a naked mole rat, maybe why the naked mole rat can inbreed is because it can accept these high levels of relatedness. And maybe that artifact was, we've all said, we actually had a laugh once. I was with Chris Fawkes. I was in a little meeting with him, and he said to me, Nigel, he said, all that work Jenny done, it could, excuse my language, he said, it could be a lot of bullshit. He said, because we caught animals that are highly inbred. He said, it could be that that's actually not the pattern that normally occurs in naked mole rats. So I said, it's possible, Chris. And he said, you know, we could actually be barking up the wrong tree. And this is where it's important, I think, to know the genetic relatedness of your animals and from the field, right from the beginning. And in those days, when Jenny done her work in the 1980s, I mean, DNA fingerprinting wasn't around.
Right.
I mean, it was still being developed by Mike Bruford was still developing it with, I can't remember the guy's name now. It was a very new coming in technique, and they used it for humans. It wasn't being used on animals. So, you know, as you say, as new techniques and advances occur, maybe we'll be able to unravel this a lot faster. But I don't think it's a simple mechanism that's taking place. I think it's complicated, and we have to be careful not to make too much. That's why I mentioned initially the high degree of relatedness, to try to bring in, this is maybe why they're able to have this control over reproduction. But it could also be that, you know, naked mole rats also may have a slight tendency, when they're not confined to a space, to outbreed. May they prefer to outbreed rather than inbreed? We don't know. I mean, it's something that has to be done in the field. And unfortunately, most of the countries where naked mole rats occur now have closed up their borders to doing research in there, because they've seen the importance of them, because they're very good models for aging, they're good models for cancer research, and they've got other very, very important possible health benefits. So countries aren't allowing you to go and work on these animals anymore. They want you to work with local people, and there's a lot of control. So yeah, I think that there's a lot more that we could delve into here. And, you know, I've touched on some things, and I can't explain everything. That's why I said at the end, we don't still know what it is. I thought I would find this answer very quickly. I thought I was going to unwrap this in a few years, and so did a lot of other people. I mean, now people are talking to me about single sequence cell work, doing this other complicated stuff to try and unravel things. I think it'll go on and on, but eventually the only way to really unravel it is to be able to learn to speak to the mole rats and ask the queen, how do you do it?
Thank you.
Pleasure.
And we have a question by Lauren.
Hello, Nigel. Thank you
Hi,
for
Lauren.
that wonderful talk and discussion here. I'm Lauren Hayes. I'm a professor at the University of Tennessee at Chattanooga, and I study more plural breeders, the degus. And I also wanted to thank you for sort of the things you've been saying about the challenges to unraveling these questions. It really speaks, I think, for the students to know that some of the stuff that we're doing doesn't take a year. You know, it takes decades to unravel. I really appreciate that message. So I apologize if I missed this, but you were talking about prolactin, and I'm looking at your figure over here where in the naked mole rats, there's a lot of variability, I think, in the prolactin. And I was just wondering, and again, I apologize if I missed this. Are there any like signatures, behavioral signatures of the animals where, you know, prolactin's associated with different behaviors or different levels of parental care in the non-breeders that you know of?
Yeah, we've just had a student, Seppi, actually gave a talk earlier I think a couple of years ago to the fine community when she was doing her PhD. And she's been looking at entire colonies, and she looked at prolactin levels. And she found that some individuals, depending on the position of the dominance hierarchy, depends on the level of prolactin that they had. And she found, for example, I think if I recall correctly, that some of the males, the intermediate sized males, had high levels of prolactin, and some of the bigger males had lower levels of prolactin, and likewise with the females. And this could be that they are less suppressed. You know, it was very elementary, because we only had 5 colonies, and it was a degree, and we had to do it within 3 years. And she had to actually validate personality in the naked mole rat. And then she had to validate a assay for measuring fecal glucocorticoids, doing ACTH challenges. And then she had to also validate assays to use a GnRH challenge to measure fecal androgens, progesterones, and estrogens. So she did that. And what we found was that the smaller animals, which do more work, seem to have higher levels of prolactin. And the larger animals, which do less work, had less levels of prolactin. So that would fit in with the cooperative breeding whole thing, and the contribution towards work. But I think what would ultimately be the answer would be to actually do an experiment. We actually did this initially. We actually had a colony, and we measured the prolactin levels in all the animals. and the steroid levels. Then we kept the queen together with the colony. The next point, we isolated the queen. We had a mesh that she couldn't be in contact with the colony members. And then we looked at the steroids and the prolactin again. And then eventually we took the queen away, and then we had all hell let loose. We had a new queen come up, and prolactin levels rose again. They were actually quite low when the queen was isolated off from the group. So the queen, through not being able to push them and shove them, the prolactin levels rose. And dropped. And then when a new animal became the queen, those prolactin levels rose again in the animals. So it looks like there is some link between prolactin and the queen's position in the colony and work and maintenance. But I don't want to say too much about that, because you've got to be incredibly careful of doing correlations and causation. So yes, smaller animals do tend to have higher levels of prolactin and they do more work. Larger animals tend to have higher steroid levels, particularly testosterone. They have lower levels of prolactin and they don't work as much. They're a bit more lazy.
Thank you. Fascinating stuff.
That's a pleasure.
Okay, I don't see any more questions. Thank you again a lot for your inspiring talk, Nigel. I'm
That's
also
a pleasure.
showing once again that when we talk about evolution, we also talk about the evolution of physiological mechanisms. As there's no more questions, I'm going to stop the live stream. It doesn't mean we have to run